I'm Liliana Silva. I'm a PhD student from Faculdade de Ciências of Universidade de Porto, and I'm developing my PhD work in CIIMAR. And today I will talk a little more about molecular evolution of olfactory receptors through jawless and jawed species. The detection of odors is extremely important for intra and interspecies communication, as also crucial for species survival. The olfactory receptors perform the major role in detecting odors, and they belong to the GPCR family, so they present conserved 7 transmembrane domains. The ligands connect with the extracellular region, and then you have the activation of G protein and the intracellular cascade. Olfactory receptors present a median size of 320 amino acids. The olfactory receptors are highly present in chordates. You can find olfactory receptors even in cephalochordates to mammalian lineage. And at the same time, the duplication events in olfactory receptors are very frequent and dynamic. So when you look to different species, we find distinct olfactory receptor repertoires. The olfactory genes were explored in some Actinopterygii species, but little is known about the gene repertoire and the molecular evolution in the majority of fish species. At the same time, there are several olfactory receptor classification systems, and they are very distinct among themselves. For example, we have one system that classifies olfactory receptor families with A to H, another one between alpha to lambda, and in the most recent to consider numerical characteristics to olfactory receptor families. The objectives of this work is access the full olfactory receptor repertoire in a sample of Cyclostomata, Chondrichthyes, Actinopterygii, and Sarcopterygii species, compare the diverse classification systems and try to homogenize them, and at the same time evaluate the molecular evolution of olfactory receptor genes across fish lineage. We start with the genome collection and the gene extraction protocol. And after these extractions, we verify that the range of olfactory receptors varies between less than 10 to more than 400 copies in some species. After our dataset reconstruction, we finish with almost 4,000 autos and a median length of 320 amino acids. So we have a saturation assessment problem here, but we performed our phylogenetic analysis considering the amino acidic sequences. The phylogenetic approach leads us to conclude that the previous reported classification systems share common points. We find the common regions and points between these 3 classification systems, but at the same time, and because you include for the first time some species in this work, you have additional unclassified sequences that belong to new olfactory receptor families. And with our phylogeny, we are able to classify these olfactory receptor families. In a general view, you obtain a good support for the majority of the nodes in our tree, but when you try to look inside each families that are collapsed here, we look the resolution, and this is because our problematic inside the phylogenetic reconstruction, you have a high number of O2s and you have a reduced length. So the next steps is try to support these results with synthetic analysis, because olfactory receptor genes usually expand in tandem duplications, and you try to assess if the synthesis is conserved across close species. Perform some DNA/DS analysis to understand the evolution of these families, and also support our our results with statistical analysis. So I would like to acknowledge the evolutionary genomic and bioinformatic groups of CIIMAR, as also the FCT to support my grant.